So, hello everyone. Thank you for making it through the ice and snow and to one of the last sessions for today in a very long day. And needless to say, I'm very excited to be here to talk to you a bit about my research at Uppsala University, and in particular about EasyMapReduce, which is a tool that I've been developing for a while now. And the idea with it is that, well, I had a problem. The problem was that bioinformaticians would come to me and they say, I have this bioinformatics tool. It works good on my laptop for a small data set, but now I have a much bigger data set and I need to scale this. And what I was doing in the beginning is that really going through a deep understanding of the problem and implement it in parallel. And as you can imagine, this is not a very agile approach because it takes a long time, and there are so many tools and so many things in bioinformatics and biology, and you can't know parallel programming, biology, and all of this stuff in detail. So I needed to develop an abstract tool that could parallelize serial tools. And this tool actually happens to work very good for bioinformatics problems. So before we dive into the details, I want to tell you a little bit about myself. I've been living in Sweden for 3 years and a half now, in Uppsala, which is a city that is a little bit northern than Stockholm. But as you can imagine and guess from my accent, I'm from Italy, actually. I've been living in Rome. I lived in Rome for 25 years. And now I'm a PhD student in Uppsala University, and I'm affiliated with the IT department and the pharmaceutical biosciences department. And my background is mainly in computer science, but nowadays a little bit in bioinformatics as well. And a strong focus of research in the pharmaceutical biosciences department is data-intensive bioinformatics. So basically how to cope with the data growth that we are observing because of high-throughput methods in biology. So for instance, drug screening, sequencing, today there were many sessions about it, and image analysis as well. And what people were doing previously, when the dataset was growing, it was buying more hardware, but now there is cloud, which is better because you can, I mean, it's more flexible. You can upscale and downscale in an agile manner. And so we are developing data-intensive bioinformatics methods on top of infrastructure as a service, and our stack is based on big data frameworks such as Spark. And we aim to offer analytics platforms and analytics services for bioinformaticians on top of this stack. And me in particular, I'm working on scaling scientific workflows. So I was telling to you before, I'm working on scaling pipelines of bioinformatics tools. So these are two main challenges that I have to cope with. So challenge number one is workflow definition. This is a whole other story. We are not going to talk about it today, but it's very important, as you can imagine. And what EasyMapReduce try to solve is isolation and parallelization of existing software. So let's start to talk about isolation. Isolation is important because all of these bioinformatics tools, they come with a bunch of libraries, and your sysadmin is going to get frustrated to install them because they conflict with each other very often. And also, the versioning of the libraries and of the tool is quite important because in science we want to be able to reproduce the results. So it's not always possible, but you know, you need to be able to have something similar for different runs, and if someone tries to reproduce it, he has to have something similar. So, and the version is of a software, it plays an important role in this. So by isolation, I mean also packaging of the software tool, so you can give it to a sysadmin and it's not gonna get frustrated when he has to install it. So what people have been doing for a while, it was using virtual machines. So if you look to the virtualization stack, you have your bare metal server, your host operating system, then you have an hypervisor, and you run your virtual machine on top of this. So you virtualize the whole stack. So you have a guest operating system, your libraries, and the applications. And as you can imagine, it takes a while to boot an operating system. So if you want to do it to analyze a small chunk of the data in parallel, it's very likely that the time of booting up the virtual machine is going to dominate over the time of processing of the chunk of data. But nowadays there are application containers, and in particular Docker, which is the standard de facto containerization engine. And the main difference here is that the container engine wraps around the ability of the operating system to run isolated subset of processes that you can see as a lightweight virtual machine. And this will dock straight to the host operating system kernel. So you don't have this booting time. It takes very little time to spin up a Docker container. And also, you can run much more Docker containers on a node than virtual machines because they are lighter. And let's talk about parallelization now. So there are two cases. Let's start with case number one. So what I call trivially parallelizable tools, and what people call, actually, it's not just me, is this typology of tools that you can run parallelly on different machines with different chunks of the data set. And if you do so, you will get the same result as running as on one single node. Okay? So in the big data community, we call this kind of operation a map operation. And Spark and HDFS, as you know, are an excellent solution to run this kind of workloads because we get locality-aware scaling and fault tolerance for free. And by the way, you don't have to use HDFS like any of the supported distributed file system works good as well. Case number two, tools that have to explore the whole data set in order to give you some insight. And of course, you can't parallelize always this kind of tools, looking at them as black boxes like I'm trying to do here. But there is at least one case in which you can do this. So if your tool performs an associative and commutative operation, you can do this trick here. You can have split number one and number two in one machine, split number 3 and number 4 on another machine, and then run this in parallel, like run tool number 5 here in the example in parallel on one machine and the other machine, and you will get an intermediate result. And then you will have, of course, a little bit of shuffling, and you can run, compute the final result on one machine. So you achieved some sort of parallelism. And in the big data world, this is called a reduce operation. And again, Spark is a great framework for this kind of workloads. So what is the idea with EasyMapReduce? The idea is that you will get your tool, you will wrap it in a Docker container, and then you will use the EasyMapReduce API to define your computation in terms of map and reduce operations. And then you submit it to a Spark cluster like any kind of other Spark application. And you let the Spark cluster orchestrate your containers in order to do the computation. And in this way, you're going to get locality-aware scheduling and fault tolerance on top of one of the distributed file systems that are supported by Spark. So pretty handy, in my opinion. And it's open source. and you're very welcome to test and contribute. Let's look at an example of usage. So a simple task in bioinformatics is the DNA GC count. So in this task, your goal is to count the G and C occurrences in a DNA string. So as you know, DNA is represented as, can be represented as text in a language of 4 letters. And yeah, in this example, the result is 4. So a pretty simple thing to do. And let's pretend we want to use Linux tools to do this. So you can do it in many other ways, but for the means of this example, this is quite nice because everyone knows this stuff. And so if you have a DNA text as input, you can use first grep in order to filter out G and C and display them on different lines. And then you can use word count minus L in order to count the lines and get the job done, okay? And let's say we want to distribute this with EasyMapReduce. So EasyMapReduce offers to you a Scala API that is built on top of the standard RDD API. So you start from an input RDD. So here is where you previously loaded your DNA text data. And then you specify a Docker image. In this case, we just need Ubuntu. And you specify a command that you use inside the image in order to process your chunk of data. And what EasyMapReduce will do for you is that it will, in the case of the map operation, it will mount the RDD record as text inside /input. So in your command, you can read it from there. And in the case of the reduce operation, it will mount it, of course, you need 2. It will mount it in input /input1 /input2. So what happens behind the scene is that EasyMapReduce write this file to a temporary directory that you can configure. And for best performance, this should point to a tempfs, so you keep data in memory, right? And then there is a default location where you are supposed to write your result. So EasyMapReduce will be able to bring it back to the Spark engine. And so if we sketch the commands very quickly, like in the map part, we are just repeating what I explained before for each line. And in the reduce part, I'm using a bash expression in order to sum the intermediate results together. So pretty simple. And now you might wonder, why do I need EasyMapReduce to do this? I can do it with the standard Scala API. Well, this is just an example. Now I'm going to show something that is quite useful. So virtual screening is an in silico method that is used in the drug development cycle. And I need to give you some background first on molecular docking. So this red monster that you see there, it's HIV protease protein. So it is a protein that is very important in the HIV life cycle. And this hole that you see in the middle of the protein, it's active, the active site of the protein. And if we dock a molecule inside this active site, we shut down this protein and the virus dies. So what you do in molecular docking is taking a protein representation and a molecule representation, and you want to test how this molecule is going to interact with the target protein. So there are 2 phases in molecular docking. Phase number 1 is called dock. So the docking phase will take the molecule, will put it inside the active site of the pose, and it's going to start to rotate it in the active site, and it's going to compute for you a pose that is going to likely happen in reality. And then you have a scoring phase where you annotate this pose with a score. And the score tells you how likely is this interaction is actually going to happen in reality. So this is molecular docking, and virtual screening is just taking this protein and run molecular docking over a large molecular library in order to find top hits, like the top scoring molecules that are going to interact with this protein. So we can take these top hits and then we can work a bit and to optimize them and to make a drug candidate out of it. So as you can imagine, this is quite challenging problem to parallelize, and I mean, if you want to do it from scratch, it's gonna take you a while to learn the chemistry and all this stuff. It's absolutely not trivial. But fortunately, there is OpenEye, which is a scientific library of tools that implements this stuff. And so there are basically two tools inside this library that are useful. One is called Fred, and is the one that does the docking. So in the map part, what we are going to do, we are going to basically take each molecule in input and run it in parallel in the different machines in order to compute the poses. So if you look at the command here, we specify the target receptor representation. This you can have in the Docker container because it's going to be always the same, and then the input, and we specify where to write the output. Pretty simple. In the reduce part, we score the poses, and we return the top 10 hits. And we are going to have 2 inputs and the usual output directory. And again, it needs the receptor as well that you can have in the Docker container. And I run this for you, so we can see if this scales good or not. And it actually has quite decent results. And I run this on a moderately sized dataset. This is a real vendor dataset that contains 100,000 molecules. And I run it on City Cloud, which is a commercial OpenStack installation. And I run it on top of Spark, standalone with HDF on the side. And I had 90% weak scaling efficiency, so pretty good. And yeah, this is quite flat, so you don't lose much by parallelizing in this way. And the CPU saturation was actually quite good as well, so between 95 and 98%. So one of my concerns, it was that firing up these Docker containers and mounting the splits from the TempFS would eat up too much time. But since virtual screening is compute intensive, it's worth it to fire up a Docker container in order to process the split of the data. So let's wrap this up. What are the advantages and the limitation of this approach? So first advantage, parallelization of tools made easy, at least in my opinion. This is a cloud-ready approach because it's built on top of Spark. And you get locality-aware scheduling and fault tolerance for free, which is good. And you don't need to bother your sysadmin to install the tools on each node. So this is quite relieving. And, but there are some limitations as well. So MapReduce doesn't apply to every use case. So if you can't do this trick that I've been doing here with the commutative and associative scoring, then you can't do this. You can't use this EasyMapReduce. And if you want to have decent scaling performance, you need the time of the chunk processing to be bigger than the container initialization. And this can be achieved in many different ways. Like, for example, in this example, I was loading multiple molecules inside one RDD records. So it will take a little bit more to process it, or you can use, if you are familiar with Spark, you can use map partition, so you can pipe the whole partition through the container, through a single container. And yeah, other two limitations are basically on configuration. So you need Docker engine on each node of your Spark cluster, and you need a tempfs mount to keep the data in memory. So we have a lot of time for a Q&A, but if you have any criticism, please bring it up, because this is nothing more than a proof of concept now, and I want to improve this, so please, don't be shy.
Okay, thank you, Marco.
Thanks.
Okay. Questions, Marco?
Thank you.
Hello. That was a good talk. Thanks. I have some questions regarding how do you serialize data between an RDD and your Docker containers?
How do I, what?
Serialize the data between your RDD and Docker
containers. Okay, so what happens is that, let me get through to the slides. So you see that there is this input file that gets mounted inside the Docker container. So what EasyMapReduce does is that it just takes the string, which is already serial, and then it writes it to a file. And then this file gets mounted inside the Docker container. So it's a pretty simple approach.
Yeah, I think like we also experimented with this approach. The trickier part you run into trickier parts when you actually are using RDD and it gets written as a string. And when you want to read it in Python, for example, and you're using pandas, for example, to parse it to a data frame, it will be very hard for pandas to determine whether a column is an int or a float.
Yeah,
there are some type conversions that happen.
Yeah, I mean, you can, what you can do is like convert it to a serial representation that is better than pandas and then convert it back. What I was doing in the beginning, I was using Unix pipes, but it was very messy to deal with them, because then you need threads and things. And it was difficult to detect errors and stuff like this. So these pipes might be another approach, but I found this one to be more handy.
So when you use the temporary file system, how does the resource management work?
So basically, the tempfs is just a standard Linux file system, so you can decide how much memory you want to give to the temp file system, and it's going to be fixed. So when I was running the virtual screening, I had 16 gigabytes of memory, and I had 1 gigabyte that was reserved for tempfs.
Any other questions for Marco? None? Okay, great. Thank you very much, Marco. Please show your appreciation. Thank you.
Thank you.